Sorry for that glitch. At the outset, I would like to thank Dr. Lily and the rest of the team for having invited me to this wonderful, well-attended conference. The topic that I'm going to be talking today is about a new drug, a relatively new drug, IMIGLIMIN, which is a mitochondrial function modulator. Now, this is a drug that is not yet FDA approved. It's approved in Japan and India, and it has been approved in India since 2022, October 2022. Some of you probably already have experience with the drug, and I don't know about how many people are using it and what is their experience with it, but I'm going to go through some of the studies and some of the mechanisms of action of this particular drug. So I do not have any disclosures for this talk. So basically, let's start with what is the relationship between mitochondrial dysfunction and type 2 diabetes in the first place. If we are trying to improve mitochondrial function, let us revisit what actually happens to the mitochondria in cases of type 2 diabetes. So you can see here that there are genetic factors and environmental factors that can both cause mitochondrial dysfunction, which we are going to try and correct with the help of a mitochondrial modulator. And what does mitochondrial dysfunction do? It causes beta cell dysfunction as well as insulin resistance. So both the defects, the basic defects of diabetes are affected because of mitochondrial dysfunction in type 2 diabetes. And if you correct these, maybe we can get there. Now, these two defects all of you know, but we are going to talk about something that is little upstream. So I'm going to talk about what happens a little upstream about with respect to these two defects that we have and how they are going to be corrected. So this seems like a busy slide, but bear with me, it's important to know this. As you can see here, glucose comes into the cell, it gets phosphorylated, and what you get is pyruvate. Pyruvate enters the TCA cycle, and once it enters the TCA cycle, ATP is generated, and the ATP that is there, you know, causes, it's the energy. Basically, mitochondria gives you that energy, and this energy depolarizes the cell membrane, and what you get is an influx of calcium, and because of that you get exocytosis of the insulin granule. So this is how insulin is produced if you go from this side to that side. What is important to understand here is that there is an interaction between the endoplasmic reticulum and the mitochondria, and there is some defect that happens at this point. So imagine if there is a defect, if there is the crosstalk between the endoplasmic reticulum and the mitochondria, if it is not going to happen in the right way, then the calcium channels are not going to open and you are not going to get exocytosis of the insulin granules. So this is one part of the story. So this fine process of entry of glucose, energy generation, which means ATP, calcium coming in, and then the insulin secretion that happen. If this main thing in the middle, which is your mitochondria, if there is a dysfunction of this, what you're going to get is impaired glucose stimulated insulin secretion. So that's your problem. So this is at the heart of the matter. I'm going to make this as easy as possible for you. This is the mitochondria, of course. If you look at the right side, and if you just divide that figure into two parts, okay? On the left side, what you see is glucose, and on the right side, what you see is fatty acids. So the basic crux of the matter, I may not go into the details of this for lack of time, but the basic crux of the matter is that the mitochondria is not, in type 2 diabetes, is not able to switch between the glucose pathway as a fuel and the fatty acids as a pathway for fuel as the need arises. So whatever fuel is presented to the cell, the mitochondria need to switch seamlessly between the glucose and the fatty acid as the need arises, and this is called metabolic flexibility. Actually, it is mitochondrial flexibility. If this flexibility is lost, then what you get is dysfunction. So one over the other, either the fatty acid is going too high or the glucose. So this is the genesis of glucotoxicity and lipotoxicity, and the inflexibility of the mitochondria to switch between these two pathways is the crux of the matter in type 2 diabetes, which we are going to try and correct. I really wish I had the time to explain this to you, but probably not. But suffice it to say here that on the one hand you have glucose coming down to pyruvate, on the other side you have fatty acids coming down to CPT1, and both these are going to enter the cell depending upon which substrate is predominant. So they are going to enter the mitochondria and get into the TCA cycle, and look at the bottom line. The bottom line is important, and that is the ETC. These are the electronic channels which are finally going to generate the ATP. Now, this ATP generation is the most important thing for the cell to carry out its duties and its work. So if you are going to make corrections, now metformin does this, if you are going to do corrections upstream somewhere at the level of the pyruvate or at the level of the